Our names are Nicole,
Olivia,
and Daniel, and we have chosen to look at this research paper. It was first published in February 2012 in the Journal of Neuroscience, and its title is Gene Co-Expression Networks in the Human Brain Identify Epigenetic Modification in Alcohol Dependence.
The researchers responsible for this paper had two hypotheses. One, there is a decrease in DNA methylation in the alcoholic brain, and two, there is an increase in H3K4 trimethylation in the alcoholic brain. The results were derived by comparing alcoholic and control regions, and statistical tests were executed to confirm the hypothesis and reject the null hypothesis. Higher expression levels endogenous retroviruses and genes with high guanine to cytosine content were associated with these hypotheses.
autopsy brain samples were obtained from the Tissue Resource Centre at the University of Sydney. Fresh frozen sections of the tissue from the central nucleus of the amygdala, basolateral amygdala, and superior frontal cortex were taken from 32 cases. 17 alcoholics and 15 matched control samples, of which there were 30 male samples and 2 female samples. Cases were matched and chosen as closely as possible to one another based upon age, gender, post-mortem intervals, and brain pH. Cases with a history of polydrug abuse were ruled out, and cases were matched accordingly to their smoking history.
The techniques used during this study include DNA microarray techniques and transcriptome profiling. A microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. DNA microarrays are microscope slides that are printed with thousands of tiny spots in defined positions, with each spot containing a known DNA sequence or gene. This was used to identify individual genes as alcohol phenotypes. Transcriptome profiles use all RNA molecules, including messenger RNA, ribosomal RNA, and transfer RNA, and other non-coding RNA produced in one or a population of cells. This was used to generate a systemic view of brain alterations associated with alcohol abuse.
The results obtained showed decrease in DNA methylation and an increase in H3K4 trimethylation in the alcoholic brain. The DNA hypermethylation is apparent within the top row of this figure. It shows the difference in the control and the alcoholic brains for all three genes presented. The standard deviation for MLT2A1 and THE1B have error bars that are not overlapping, which concludes that they are statistically different.
The LTR8, however, does have overlapping error bars. Therefore, a t-test was executed to obtain a p-value that can be used to dismiss or accept the null hypothesis. As the value is greater than 0.05, the null hypothesis has to be accepted in this case.
The bottom row of this figure shows mono, di, and trimethylation of the histone H3K4 and are compared by testing the control brains against the alcoholic brains. We can observe that there is a greater trimethylation. To confirm the significance of the results, a t-test was also conducted and the p-value result was less than 0.05. We can therefore reject the null hypothesis. The genes that were identified by the microarray technique were confirmed to be alcohol phenotypes. These genes were specifically tested for the degrees of trimethylation and were represented by this figure. It shows that there is a significant increase in H3K4 trimethylation in the alcoholic brain. This can be further supported as all three genes possess a p-value lower than 0.05.
The hypothesis was tested to see if DNA in brains of alcoholics are less methylated. This was confirmed by the previous figure. Hypomethylation results in transcriptional human endogenous retroviruses and high expression of guanine to cytosine content. These results were displayed in a module network which was designed to show overlapping genes and co-expression modules in the human brain.
All modules situated within a dotted square box have a high density of co-expression modules. The connecting lines between individual genes indicates a significant overlap of genes between modules of different brain regions. The alcoholic effect on individual genes is demonstrated by the color differences. Where an abuse of alcohol consumption is present, gene nodes that are highlighted in yellow and orange are upregulated, whereas the genes that are represented with blue or gray are downregulated. The greater intensity of the color, the greater the effect of the up or down regulation. T-tests were conducted for every transcript in each brain region to compare gene expression between alcoholics and control cases.
There are three main findings. Number one, Chronic alcohol abuse differentially affected major cell types in the brain. The transcript modules from neuronal modules were mainly downregulated in alcoholics, while several modules representing microglia are upregulated. Number 2. Alcohol abuse resulted in upregulation of long-term repeat retrotransposons. Number 3. Most genes from GC-rich modules were upregulated in alcohol abusers, whilst genes from GC-poor modules were mainly downregulated.
As stated before, hypomethylation caused by chronic alcohol abuse also results in a higher GC content in genes, as these genes are mainly upregulated. A high concentration of these may cause chromatin modification. This concludes that alcoholic abuse may have an epigenetic effect in the genes.
As a summary of these results, there are higher expression levels of the endogenous retroviruses in alcoholic brains.
The increase of GC content in genes in alcoholic brains were associated with DNA hypomethylation.
There is increased histone H3K4 trimethylation, suggesting a role of epigenetic mechanisms in alcohol addiction.
In summary, it can be stated that the chromatin modification may be the cause for long-term alcohol dependence.
A flowchart was constructed to summarize the study's findings. In conclusion, alcohol abuse alters epigenetic modifications in gene co-expression, resulting in alcohol dependence.